From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 1st, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 90th episode of Matters Microbial. 10 more to go before reaching 100 podcasts. I am truly grateful for every download, listen, or view. I've just returned from a trip to the University of Texas, Austin, where I gave a seminar about microbial centrism, put on a LuxArt workshop, and had a remote podcast session. The folks at Austin were uniformly wonderful to me and so much appreciated. The LuxArt session was particularly impressive. Joseph, seen here, was the student who invited me as an undergraduate ASM chapter member. Chloe, seen here, was instrumental in the logistics of my visit. And Dr. John Partridge, seen here, was the ebullient faculty member shepherding me around as he talked about Nick Cage movies. As always, the creativity of the students simply amazed me. Artwork ranged from anime, seen here. Bunnies, seen here. What I can only guess is elvish, seen here. As what I often see, flowers, seen here. And a Pokemon figure, of course. Keep in mind that these are painted with bioluminescent bacteria with a brush on nutrient media developed overnight and photographed by yours truly. One student created a fabulous image of a type 6 secretory system, seen here. And another student tried to be a little bit punny, as you can see here, with a description of picking the right single colony for Valentine's Day. I'm going to be creating a video of the many pieces of creativity from this session. It was a wonderful experience, and I'm most grateful once again. In several prior episodes of Matters Microbial, we've explored the role that microbiomes can have on organisms in general or specific. So it should not be a surprise that plants have their own microbiome, as does the soil. If you need to appreciate what goes on in soil and why you should care, I highly recommend Dr. Joe Handelsman's recent book, A World Without Soil, seen here and linked in the show notes. You'll never call it dirt again. What microbes do in the soil has always fascinated me. Most of my own PhD was spent studying the rhizobium-legume symbiosis, which is intricate, beautiful, and necessary to agriculture. Here is a figure depicting its complexity, and I will post a link in the show notes. Just as we can propose to improve human health by studying and perhaps modulating the gut microbiome, it should not be surprising to think the same could be done for plants and agriculture itself. So I'm happy to welcome Dr. Francisco Dini Andreote of Penn State to the podcast to discuss the work of his research team exploring, among other things, the rhizosphere of plants as well as many related topics. Welcome to the Quality Quorum, Francisco. I'm happy to meet you.
Thank you so much, Mark. It's a pleasure to be here. Actually, I'm following along and with views of your channel and your podcast. And when I received an email from you, was a surprise, a nice surprise, and I was truly happy to be invited to be part of this show. Thank you.
This adventure for me, let's put it that way, and hopefully useful to others. I tell myself that anyway. I have a quick question for you before we begin. You're part of this very expansive microbiome initiative at Penn State. Could you say a few words about it, because it's seemingly everywhere, and I will put a link in the show notes.
Absolutely. Yeah, I'm part actually of the steering committee of the One Health Microbiome Center at Penn State. We have Professor Seth Bordenstein as our director. And it actually, I was hired at Penn State as a part of a cohort of faculty members that was hired to join the Microbiome Center. Today, it spans all the campuses that we have at Penn State. Over, I believe, 500 people are involved. We have seminar series, we have partnerships with companies, we have support to students in terms of fellowship, training. We have core facilities that support our activities. It's growing faster than we could imagine, and we are all super excited to be at Penn State at this time to work with microbiome science.
Again, I'm going to put a link to this whole initiative in the show notes. There must be a German word for a combination of jealousy and excitement, because that's what I feel whenever I hear about this program.
That's a great point. Yeah, yeah, and
Yeah.
that's something I use as well to motivate you, right? One thing I learned back in the day is if you are unsure about your career path, try to look for those things that, exactly as you said, makes you feel jealous and excited about it. Most likely, that's where your career should go.
That's so well stated. I'm going to create a meme with you saying that.
Yeah, that's okay for me. Yeah.
So I think a good thing for you to do for listeners and viewers is once again, we've talked about microbiomes before. Sometimes we do a deep dive in how you analyze it. Sometimes we talk about the relationship of the microbiome to certain human diseases. Sometimes we focus on the fact that we're analyzing poop, all that kind of stuff. But you come at it from a different direction, and I think it would be great to situate listeners and viewers.
Sure. Yeah. Well, microbiomes are the foundation of life, I would say, right? So if you think about the history of the Earth systems and how the atmosphere started to be oxygenated billions of years ago, we talk about the oxygenic photosynthesis in ancestor cyanobacteria. Then later on, which we can move on when we talk about plant microbe interactions, there was this kind of seminal transition of aquatic plants from either saline water or fresh water towards like terrestrial systems. And now we envision that that transition was only possible because those ancestor plants, they early on associate with microbes in the soil. And the argument behind this is when an aquatic plant needs to move toward the terrestrial system, they need to cope with drought stress. Well, there's lack of water. They don't know how to regulate that. And they need to cope with the absence of nutrients. And then this early association, specifically in this case with arbuscular fungi, or
Yeah.
AMFs, was critical to kind of expand the root systems and allow these plants to really stand in the soil and explore the soil as kind of a substrate for nutrient uptake. And then if you say, well, which example or which validation we have for that. I like to give this number to some students. If you think about AMF and plants, about 80% of all the plants we know are able to associate with AMF. And then what's even more shocking is if you enjoy flowers, right? And the angiosperms, they have over 300,000 species. And you can even argue that this radiation of the evolution of terrestrial plants were only possible because of this early association with AMF. And I'm just giving one example. You talk about nitrogen fixing bacteria. You can think about many other microbes. So microbiome is basically this collection of microbes and the environment they live in, and are essential for everything we see in terms of life on this planet.
It's interesting. When I studied the Cynorhizobium mellilodi and alfalfa, one thing that I thought was interesting is we didn't spend a lot of time then discussing how the specific rhizobium cell was able to find the root hair. Because it's not in, I mean, we would sometimes do them in tubes on plant culture media, right? But that's not what happens in the soil at all. And I used to sit and ponder about this. I don't know what to call it, the extremely biodiverse rhizosphere, the number of microbes and bacteria, archaea, fungi, and eukaryotes that are in that soil. It's roiling with life. And yet this plant has to be able to somehow attract this one type of bacterium, and that bacterium has to find it. So it's the crosstalk I found so fascinating in this. And I'm not saying it's the same as we find, for example, in the human gut, but it may be actually far more complex.
I completely agree with you. So that's the forefront of plant microbe interaction science at the moment. We call this chemical communication between plants, microbes, and sometimes between plants and plants. So that can be mediated by what we call exometabolites, like something that the cell can produce around their cells. But you can also think about what we've been studying a lot, volatiles. So volatiles are those molecules that can be really diffused through the air, right? So if you think about the soil as something that contains airspace and some aggregates, and then you have pore space for water. So if you do have like a lot of like these molecules that can permeate or be diffused through those patches, that's a very important way to communicate. And plants can produce exudates, but they do produce volatiles, and microbes can produce exometabolites, but can also produce volatiles. So those chemical intricacies is something that we study a lot in terms of plant attracting microbes and microbes also modifying plant root traits sometimes.
I think it's interesting you bring this up because most everybody has the experience of the fact that one volatile, ethylene, is a ripening agent in plants. And this is an issue with your bananas at home if you keep them in a bag. You know that, and that you think, well, okay, that's just in the bag, and then you think, wait a minute, this takes place in nature, and there's all these different things happening all at once. It's as complex as any biodiverse type of environment you could ever guess.
Yeah, and another example I give in class is the geosmin, right? The petrichor. So that is something that's produced at a very tiny amount into the soil. And we as humans, we evolved to perceive that, the smell of the rain, right? And actually, it was evolution associated with our perception of whether or not a meat is proper to be consumed or not. And then we associate with that what are volatiles, and then basically you have the rain permeate in the soil. So this air space that I mentioned was occupied by a very tiny amount of geosmin, and then the water takes that place, and the geosmin gets out of the soil. We smell, and you have this feeling of, wow, that's the smell of the rain, right? And that's the geosmin produced by bacteria in the soil. So
You
that's
know,
another example of chemical communication. Yeah.
one of my favorite demos to do in my microbiology classes, I'll have a streak plate with streptomyces, and I'll have them waft it, and they always say that it smells like dirt, even though we don't like that word. It's potting soil,
Yeah.
thank you. And then I'll have everyone turn and say, no, dirt smells like streptomyces. And this is the thing we don't think about. There's this constant microbial communication taking place everywhere. We only perceive a tiny bit of it.
Yeah, absolutely. And just going back a little bit more towards the science, we do work with specific exudates, right? So we work with specific flavonoids that can activate the attraction of specific bacteria. When I talked about AMF before, we know or we understand now that strigolactone's exudation is a major component of attraction and branching in arbuscular mycorrhizae fungi. Uh, and that's something that actually is induced by the lack of phosphorus in soil. So
Ah,
strigolactone is produced, but if there's lack of phosphorus, then let's produce a little bit more. Let's try to communicate with those AMF. And yeah, and that's an active area of research for sure.
so it's funny. You think you're outside, you think it's a quiet summer day. Well, to you, but everything's talking.
Yep, absolutely. Yeah. And it's fascinating how this kind of ecological interactions takes place, and it's so context dependent, right? And later on we can get into this conversation, but strigolactones is also molecules that induce germination in a parasitic plant.
Oh,
uh,
we'll get there when you need to. Absolutely.
yeah,
I'm nerding out with you right now, or I guess
yeah,
geeking out. Although the geek and nerd thing always confuses me, but I'll take both. Um,
I love that.
Perhaps we should go ahead and just talk about some of the research that you're doing in your lab then.
Yeah. So well, I can talk a bit about my trajectory. So I'm agronomist, but since my first years in agronomy, I was never into farm and food production, but I was geeking in the lab. So I started my career as a molecular biologist in the lab, and then I transitioned that to molecular microbiology. Then I did a master's in genetics and plant microbe interaction. And but later on in a PhD, I moved towards the field of environmental microbiology and community ecology, which also fascinates me. And then once I moved to Penn State, I'll start to work with plant microbe interactions again. And just to put into context, if you ask me how I run my group now, I have all these layers of science compiled in each of my students. Exactly. So I have students working precisely on plant microbe interactions when we look into exactly what you are talking about, like root exudates, chemical communication. I have another layer of research which we try to look into the root as a niche for different microbial species, even though the root's not a niche, but is actually provides resource that defines the niche of different species. I like to show, perhaps I can put in a slide now, slide number 8. So that's a very foundational definition of what ecological niche is. So the idea behind that is once we start to understand how organisms are distributed in the environment, there is this fundamental question of what is ecological community, right? And initially they thought, well, all the organisms are interacting in this ecosystem, and this super organism, which is composed of many species, they act together. Later on, people said, well, maybe that's not a very kind of precise definition. What happens is there's different resources in the environment and they fluctuate over time. And then different species, they respond differently to those resources that are available. And then Hutchinson later on came about and say, well, if you think about any dimensional space in which each axis of this space is a resource, then everything that fits within that, it's a niche for a species, right? And I like to envision the rhizosphere as a niche as well. So the root of the plants as a niche, how they modulate resources that affect organisms.
And it's interesting because the inhabitants are modifying the environment, the niche, as well as the reverse. I mean, it really is like a handshake with the environment and the organisms in it. And it works in both directions, which fascinates me no end.
Exactly. And if you go in that direction, we can talk for hours and hours about foundations of ecology, right? Because then we talked about priority effects. So it means the first organism that gets into an ecosystem, as you mentioned, they modify the environment. And how that affects when the second species comes in, that modifies in a different way. So the order of arrival may affect the fate or the outcome of the community. And if you think about Rhizosphere, it makes all the sense, right?
And then I think about the agriculturists that want to add particular bacteria to the seeds when they plant them. And I'm going, and then they have to think about competitiveness factors. I mean, this is it to me, it's interesting because we tend to look at the soil as being this simple thing, and it's just as complex as any rainforest you've ever seen.
Oh, it's more. I would say it's more. Yeah, I use this analogy in a classroom. So if you think about number of species and all the intricacies, and you mentioned things in there, you said, well, it's not only bacteria, it's bacteria, archaea, and it's protists, it's fungi, and on top of that, you have microinvertebrates, you have nematodes, and all the trophic interactions that's going on. It's for sure the most biodiverse ecosystem on Earth and the most important one, right? So if you think about the regulation of our climate is defined by them. If you think about the food we eat is defined by them. So all the One Health concepts that we discuss nowadays, as in the center, it starts in the soil.
I agree completely. And it's funny because, well, I'm getting off track, but I guess we can lead to some of the things that kind of impact agriculture that you wanted to lead up to, or did you have another project you wanted to discuss first?
Yeah, we can just continue with those area of research as I was saying. So basically, my lab nowadays we work with chemical communication. We work with how microbes perceive these resources in the rhizosphere. We work on how these resources shift in terms of biotic and abiotic stressors like drought, nutrient deficiency, salinity, and sometimes combined effects, right? So how drought and the addition of microplastic in soil interacts. So in those cases, sometimes we are really interested in understanding how stressors interact and how that interaction affects organismal interactions. And on the other end of the spectrum, I also have some students working foundations of community ecology. And I'll keep that very short. But what we try to do is to use microbes as experimental units to test ecological principles or hypotheses that we cannot do in plant systems. right? And that's fascinates me as well. Just giving like the how easy it is to manipulate microbial communities and see outcomes compared to trying to run those experiments in plants or animal systems.
This reminds me a little bit, and I'm sure you've seen it, and it's a very simple version of what you're discussing, but I think it may be relevant to listeners and viewers. The way that sauerkraut develops is that there's all these ecological successions. In fact, I'm going to submit a figure of that process that you're looking at now. And what's interesting about this is that one organism seems to prepare the way for the next. And that's not quite the same as what you're saying, but it reminds people of the interactions and complexities, even as something as simple as sauerkraut or kimchi.
Absolutely. Yeah. I did my PhD looking into ecological succession of organisms in soil, and you can really track that. And sourdough is a good example, but you can think about many other aspects of your gut system, right? So let's say you take a treatment with antibiotics. So in most cases, if you consider treatments like a fecal microbiota transplantation, case of some specific disease, they see, oh, there is a higher integration of this microbiota. They call that engraftment after a treatment with antibiotic. And that gives me the room to introduce something we study here, which is mixing of two communities,
Mm-hmm.
right? Or invasion of not a single organism, but multiple communities, in which in ecology or in microbiology nowadays, we call microbial community coalescence,
Yes.
right? So I can even perhaps illustrate that with slide number 5. So that's an area of research that go in line with what you said about this order of arrival, that which is ecological succession. We can also use other community ecology principles, in this case, invasion biology, but slightly different. So here in this article specifically, we make this kind of a comparison between principles of invasion biology as a framework and principles of community coalescence, which is mixing of two communities as a framework. And it's very interesting to look at this because if I say to any listener now, think about two forests, like a tropical forest and a temperate forest, getting all these organisms, mixing them together, considering all the ecosystem variables that they have in there. What would be the outcome? That's kind of crazy. It's like, why should I think about it? But then if I say, do you shake hands with someone? Do you hug some person? Do you have animals? Like, do you pet your animals? Or in case of this article, I even asked my postdoc at the time, like, can you take a picture of your beautiful daughter with your dog? And he put some peanut butter in her face. That's the image you can see in there. And the dog is licking her face. So basically, we have the entire microbiome or microbiota of one organism mixing with another. We see that in coastal areas. I did my PhD in salt marshes, right? So you have this flooding of the seawater coming into a terrestrial system. And the question I try to answer is, what's the outcome microbiome once you have this mixing? And how can we ecologically parameterize this mixing? How can we advance this field of community coalescence as a field of invasion biology that's more realistic, it shows what we have in microbial systems?
Oh, that's really cool. I have a friend, Dr. Maya Breitbart, who studies all kinds of aspects of environmental virology, and she's this is very harsh, I know, but I understand the spirit. She says specialization is for losers. And it's not really true. Please don't get angry, listeners and viewers. But the fact of the matter is, when you have a lot of interests, if there's a way you can address them, look at the joy it brings you. Seriously.
Absolutely. And it's all integrated, right? So if
Yes.
you say, Francisco, how can you integrate that into agricultural science? Like, well, we apply manure in the system very often, right? And what you're doing is we are making a coalescence of the manure microbiome with the soil. And then if you say, well, we use biostimulants or bioinoculants, and a major component you already mentioned is, well, they survive for a short period of time. So what really determines the capacity of invader to enter in the system? So these fundamental aspects of community ecology is tightly connected with our capacity to more precisely manipulate microbiomes in the clinics, for example, but in my case, in agricultural settings.
That's really fabulous. And it's important because many people I run into, and I guess this is, I hate saying this, but our current climate is that people are looking with a very jaundiced eye at science. But you see, as you just mentioned, the things that you're discussing are not removed from things that we need to know about every day. And I think some folks don't appreciate that. And so I'm glad to hear that you can relate this to agriculture itself.
Yeah, that's my primary focus. I guess what put into perspective, I believe there is a lot of agricultural science in which we manipulate the environment. As I said, we apply manure, we reduce tillage, we put mulch, we do a lot of agricultural management in there that we see benefits for the crop production. But in essence, most of those managements, the heart of that is the soil microbiome,
Yes.
right? So if your manure is being decomposed and mineralized, microbes are doing that. So if your mulch is staying a little bit longer because of the C, we call that a C/N ratio in the soil, whether a substrate has a labile kind of degradation or if they are more recalcitrant, microbes perceive that and they determine the fate, the modification, the stabilization of organic matter. So integrating this kind of, it's not interdisciplinary, even though I often think as it is, of what is a practical management in the system and what are the core foundation of knowledge of what's happening in terms of the biology of the system is something I try to connect every day.
Wonderful. So, a specific project that you'd like to discuss?
I can talk about perhaps the Striga if you're excited about it. We could talk about it.
Well, most people, I mean, I knew about it because when I was in graduate school, a fellow graduate student had worked on it in Virginia as an undergraduate researcher and was petrified of Striga, how tiny the seeds were. So maybe you should talk about Striga and what it affects and what it impacts. I believe it is quite a problem in Africa.
Yeah. So you can put like slide number 4. So in summary, so Striga is a hemiparasite. It's a parasitic weed. So basically, it's a plant that parasitize another plant, right? And in this case, there is over 50 million hectares in sub-Saharan Africa, over 40 countries has been impacted by Striga. Uh, and in those regions, it's really hard to control that, even though you have agricultural management, as I explained, like push-pull systems, or even like removing that by hand or trying to breed for resistant varieties. But in most cases, that's not like sufficient. And the way this parasite kind of infests the field is that a single plant, they need for germination specifically. So those seeds are very tiny. You can think about that as grain of sand. Uh, and to germinate, they need a period that's called preconditioning. So they need to stay in the soil under a warm, kind of wet condition for a period of time. But most importantly, they need to perceive a signal of a host plant. And that signal is strigolactones, right? Think about it, in the absence of phosphorus, plants exudate those molecules. And then this parasite evolved to have around 11 different receptors in the membrane to perceive strigolactones. There's papers showing that how they repurpose these receptors from one kind of function to be a receptor of strigolactones. And once they perceive that, so they initiate the internal kind of ethylene biosynthesis, and then they produce a radical. So the seed starts to germinate. So the next step of the infection is to develop what's called in parasitic plants haustorium. So I like to think about haustorium as a mouth, but it's basically this kind of tip of the radical that allows this parasitic plant to really connect to the host plant. And once they connect, they do what we call a xylem-xylem connection. And this parasite starts to siphon nutrients from the host plant. In this case, can be a cash crop like sorghum. And by doing that, then they start to grow out of the soil. Once they get out of the soil, then they can do photosynthesis and produce thousands and thousands of seeds again. So this is kind of the life cycle of Striga. And then I was involved in a project with two amazing grad students. And even though the project have some kind of directions in what you should do, I really enjoy like talking to the students about how do we envision that. So over coffee, maybe you can put slide number 6. We develop this kind of synthesis of how potentially soil microbes can control Striga.
Oh,
right? This figure is a little bit complicated, but to put in a nutshell, I asked him, I was like, how do you envision that? And he said, well, there might be ways that microbes can solubilize phosphorus, give nutrient to the plant. And in doing that, the plant may exudate less strigolactones. Like, great, phosphate solubilizing bacteria. That's a good point. Let's call that indirect effect, because basically you are helping the plant to fight against the striga. What else? And then we discuss those ideas, right? And other ideas would be, what if some microbes directly affect Striga, right? They produce some chemicals that kills, or they parasitize, or that they are pathogens of Striga seeds. And I call that direct modes of action. So microbes directly controlling the Striga seeds in the soil. And what fascinates us in this project, which is still ongoing, is right now, if you remember this story of Striga germination, we identify some microbial volatiles that once you expose the seeds to those volatiles for a given period of time, later on, if you give the strigolactones and try to germinate the seeds, they remain dormant. So now we are working towards understanding how this is happening and scaling up like field trials to use those microbial volatiles, actually use a molecule that induces the expression of those volatiles to suppress Striga germination in soil. So that's really fascinating. It's really like chemical communication, and it starts a simple microbial kind of type of assays in the lab, and now we are scaling up that to soils.
my guess is have you found that these are single microbes that are producing these, or are they doing it as groups?
Yeah. So what we found is we found single strains in the lab that can do that. We identified the metabolite, we identified the precursor molecule, and when you add this precursor molecule in the soil, a lot of microbes produce that. Even microbes that, or in those that we identify as strains, we cannot detect in the soil. So it's a trait that's kind of spread across multiple microbes. And we did all type of validation. So we gamma radiate the soil, add these molecules, there's no control. So we need the microbes to be there. We did metatranscriptomics in the soil, and you see the expression of the genes for the production of those volatiles. So it's really a microbial mediated mechanism. And that's something that fascinates me, because most of our view of manipulating agricultural microbiomes still comes from the idea we need to cultivate a bunch of that into a container and put that into the soil, right? And I have a critical view on that, especially from ecological perspective, because ecologically makes no sense, right? That we are replicating only a single strain and putting that into the system. But in this case, we are just turning on and turning off a mechanism of that the function is already existing in the system.
You know, when I was in graduate school and we would study nodulation, we would sprout alfalfa seeds on solidified tissue culture media. We surface sterilized the seeds, for what that's
Mhm.
worth. And then we would add cynorhizobium in the tube. And I used to say, well, this is nothing like nature. I understand that you have to have those reductionist approaches when you're first studying mechanisms and you build from that. And in fact, the field has done that in the people who study rhizobial interactions. But when it was first being done, they start that way. So I want to give proper credit to that. But I'm not at all surprised to learn that there's this web of interaction that takes place everywhere.
Absolutely. I completely agree. I think reductionist approach is what you said is important for understanding mechanisms, but it comes with some intrinsic bias sometimes. Analogy I like to make is if you look back into history of our disciplines, right? So I had this conversation with, it was a mentor for me, even though I'd never worked directly with him, but we have a good conversation, Professor Brandon Bohannon.
Yes.
Uh, and one time he mentioned to me, he said, do you ever realize that most of microbiology, when they emerged, if you think about Pasteur or Koch, the work they did, they come from a background of chemistry. So chemistry is around 100 or 200 years earlier than the field of microbiology. And in chemistry was a very common approach, try to identify chemical compounds, isolate and purify them, and test concentration dependent effects, right? I'm oversimplifying chemistry here. Uh, but that's kind of the line of reasoning that you have in
Mm-hmm.
mind when you think about the roots of microbiology. We try to, that's what Koch did, right? So if you see a pathogen, let's take the pathogen out, be able to cultivate them, And then you need to infect again and prove that's the pathogen. Uh, and when you think about ecology, on the other hand, that's a little bit more complex, as you said. We look into the complexity of nature, and if you take a piece out, nature will rearrange, right? Or if you put more, you cause a biotic impact. So it's a slightly different view of the systems for sure.
Oh no, that's a 100% true, and I think that Brendan's quite right in what he was saying. What a great thinker he is, by the way. It's been many years since I've spoken with
Absolutely.
him. But this business of reductionism in the early days of microbiology, I would also add that they were fighting the germ theory at the time, fighting for the germ theory, I should say, against
Yeah,
spontaneous generation. That was part of
absolutely.
it. And the medical aspects and veterinary aspects were driving their interests. And that's where you get single colonies and pure cultures when neither of those things occur in nature. It's mostly interactions.
Yeah.
But if you think about it, microbial ecology originally we didn't have the tools to really dissect things, but now we do.
Exactly, exactly. That's how I see it. There is a lot of value on that. I guess my critical world is just like, can we be more creative in the way we manipulate microbiomes, right? So
Yeah.
especially in agriculture, we try to find beneficial microbes, but then we need to be able to first culture them. We need to be able to produce that at a larger scale. Then adds to that what's called a shelf life, right? So if you can find a bacillus or bacillus, that's better. They can sporulate, right? We don't want to work with organisms that we cannot store, right? And all those things are based on this principle of inoculation, right?
Yes.
One figure I like to show in my lecture, maybe you can pull off slide number one. Uh, I use this as an example. So these are average numbers. And I use this example of from a plant breeder perspective, right? So you work with genes, alleles, etc. But then you have this kind of plant genome in which you can try to modify one way or another to get a specific trait that you are desired, right? That you desired. It can be like tolerance to stress, or it can be tolerance to pathogen, or it can be nutrient in the grain, or productivity, right? When you look into like the microbiome around the plant, and here I'm not putting viruses, I'm just adding like bacteria, archaea, fungi, and protists. So similar to our gut system, that's an extension of the plant genome, right? In terms of metabolic potential. And what we're trying to do here is to find a single strain that's very good, over multiply that and add it back. So most likely the function's already there, right? So would not be more smart to consider and say, can we activate the function we desire? Can we make that organism that has that specific function to create an environment or activate or make that function to occur in that time that we desire? That's a more complicated question. I completely get it, but might be a more ecologically realistic scenario in which you can manipulate or engineer microbiomes.
You know, this is two things very quickly for listeners and viewers. With all due respect to certain manufacturers, when you take a probiotic capsule, the question is how many times that actually ends up in your lower GI tract, and that's a very different question than what's in the pill. And that's what you're saying is that when you put inoculants into the soil, the question is, do they persist? Do they interact correctly? And we don't think like that. We just think about dumping it into the soil. Rather, like I was doing nodulation assays on alfalfa all those years ago. In addition, I just want for listeners and viewers to remember that this all might sound like nerding out, whatever the term is you want to have over this complexity. But currently, how we deal with certain pests in agriculture is to douse them in herbicide. And I would argue that this is a much more elegant and effective approach and less damaging to the environment.
Yeah, yeah, we talked about that very, very, very often. In terms of you mentioned about taking the capsule, whether or not it works, or binoculars the field, whether or not it works. There are cases of success. I don't want to
Mm-hmm.
discredit that. That's very context dependent in terms of how nutrient rich is your soil, which climate you're operating, and for how long you expect that function to occur. Right. In terms of agriculture, I like to say that we know how to manipulate soil chemistry. Right. We apply
Mm-hmm.
limestone to correct pH. We apply nutrients. We apply fertilizers. The next layer is how can we manipulate soil life, right? And that's critical for the goal that we have, which is called sustainable intensification, which means we need to double the amount of food we produce in the next 30 years or so uh, without causing more damage to the environment, right? And how can we do that? I believe that microbiology is the next frontier, right? So exploring those microbes to have those functions activate at the moment we need, either to control a pathogen or to enhance nutrient uptake, to manipulate plant roots, to solubilize nutrients. That's the frontier that we are doing progress, but it's still where we can expand more, right?
This reminds me a little bit of what the great Norman Borlaug said about his quote, green revolution. When you listen to him talk, there's a wonderful video I'll put in the show notes. He says, you have to listen to the soil, you have to listen to the plants. And he doesn't mean quite the way you and I mean it, but he's exactly right. It isn't independent of nature. It is part of nature, even though, yes, we have disturbed it to grow our crops in it. And we tend to forget that.
Yeah. And in terms of agriculture, I can speak that farmers we sometimes have this negative connotation or negative view and say, oh, there's extensive use of pesticides, right? But the farmers are struggling to produce the food we consume. We should be
Yes.
very kind of, grateful to that. And they are often very open to alternative strategies, right? So I can perhaps mention another project which is not under my leadership, but I do collaborate here on campus in which we apply what's called anaerobic soil disinfestation. I don't like the word disinfestation, but that comes from back in the day in the vegetable production. They used to fumigate the soil and
Yes.
basically kill everything, right? To eliminate soil-borne pathogens. So what this technology is proposing, and I already said it's not disinfestation, is that we add organic amendment into the planting system. We cover that with a mulch plastic, which is not sustainable. It's the use of plastic, something we are discussing. And we flood that system, right, with water. Basically, what we're doing there in simple terms, we are fermenting or composting organic matter on site. And once you do that, you control for a lot of the soil-borne pathogens. You increase the nutrient status of that soil, and it's all entirely a microbial process. You see it going from aerobic to facultative anaerobe, all the anaerobicity, and then coming back into the normal state. And we're doing even studies prior and after, and you see a recovery of the soil microbiome. There's no impact on that in the long term. And we are controlling for soil-borne pathogens without using any chemicals, and we are enhancing nutrient without using external inputs. So that's one example of sustainable intensification, reducing the external inputs in the system, the damage to the environment, and just producing quality food. And we do that in an organic system. So just to put into context. And then again, this is an agricultural thing. A lot of farmers in Florida and in Pennsylvania are using that nowadays. And it's essentially a microbial driven process. Everything is mediated by them. Yeah.
Well, I mean, my nickname about microbiology is first evolved, last extinct. I also call them the small masters because they are. I have a tattoo of that actually. Anyway, the reason I bring all this up with you is reminds me when I was a little boy, my father's father was a farmer, and I was very anti-pesticides. Oh, it's a terrible thing, worst thing in the world. And I was maybe 8 or 9 years old, and he's sitting on the back porch with me, and my dad didn't have any tolerance. My grandfather, probably my age now, by the way, he was saying to me, well, if you don't like pesticides, you have to plant about 4 times as much. I said, what? He says, well, yes. I mean, the insects are going to eat it and other types of pests, and you have to watch it carefully. But then what's your goal? You need to make food. And it really took me aback. He's not wrong in what he says, and you do have to balance the other aspects. And in my experience, as you say, farmers are happy not to do, all the pesticide stuff and herbicide stuff. They would much rather have another way to do it.
Yeah, absolutely. Those are inherent discussions we have in agricultural science, right? For example, I'm generalizing here, but usually if you make a conversion from non-organic to organic system, you may expect a reduction in the production in your system, right? That's expected for a short period of time. And then you can even argue saying, okay, but then you can charge a little bit more for your organic products, and then you can compensate the profit. But if you think about the total volume that's produced, that means that you need larger area to produce the same amount of food. So which also can impact the environment. So
Hmm.
all these discussions or this context should be kind of integrated, and even in terms of management strategies, I mentioned about the anaerobic soil disinfestation, which say, oh, that's great, let's do that. Our concern now is first the use of plastic. The second is, well, we are fermenting or composting organic matter into the soil. How that affects greenhouse gas emission. So everything is tightly connected. Even though we try to be more sustainable, there's always room for improvement and for things to be considered, right? When you think about no-till systems, you say, I'm not applying tillage, which is like revolving the soil, then I maintain soil health and structure. But then you have problems with weeds, then you need to spray more. So all these nuances there's not a single answer, right? And that's the beauty of agricultural science. So how we navigate that system that also varies across seasons and regions and climates and cropping systems, and how we work every single day towards optimizing those strategies.
When I was a boy, my father insisted we grow in our backyard tomato plants and strawberries, things that don't take up a lot of space, just to remind us of where food comes from. And I think a lot of people, and I'm not judging anybody, sometimes they don't appreciate the process at all. And so maybe it would be good for everyone to do a little bit of that, just to see, grow some herbs, just
Yeah,
see, right?
yeah. I call that connecting the society with the food system, right?
Yeah.
So a review published back in the day showed that while we can be more sustainable, we need to increase food production, but a major call was food waste. Actually,
Yes.
a lot of this sustainable aspect goes from we buying cheap stuff or things that does not cost a lot, we don't consume and we put that in the trash. And that happens in our house, right? So if you could cut food waste, we would be way better in terms of food supply overall. And that is, in my view, is what you said. Sometimes people lack this connection with the food system. So if you ask what is a food, they say something I grab in a supermarket, right? Or you and I agree that that connection is super important. As an example, when I was living in the Netherlands for 10 years, they have this community effort that you could rent a space and you and your neighbors could go there and plant and put some stuff in there. And when you ask, like, why are you planting those things? Like, oh, just to get my kids connected with the land.
Hmm.
And that's super important. It changes your perspective of how you value food. When you're eating and you have one more bite and say, maybe I'll throw that in the trash. Like, not really, especially if you're eating some products like meat or something that takes way more energy to get into your plate.
That's
So
correct.
kind of elevating these values into the society is for sure critical. I teach that in my agroecology course.
Yes, you actually made a comment of this on your website, agroecology, and I really love that. Would you give listeners and viewers a definition of that term, because I find it lovely?
Yeah. Well, when I studied agronomy, everything was agriculture, right? And agriculture, in most cases, you think about the production per area and the profit and how you optimize crop production to have profit. So usually this view of profit is your goal, right? When you move into agroecology, we integrate the field of ecology and agriculture. And in simple terms, I love this definition. Ecology is the science that study how nature works, right?
Ah.
Uh, so the analogy I make in class, if you go into a forest, I say, how much external inputs needed for a forest to persist over time? They say none. So you have internal self-regulating mechanism, right? So species are competing, interacting, and then you have the nutrient cycling dynamics happening. So it's a self kind of sustainable ecosystem. And then when you think about agricultural system, so we are harvesting, we take things out. So essentially need to put some energy back. But how can we envision this system to be more sustainable by translating principles of ecology into agricultural science? So that's basically what agroecology is. You can go all the way from complex permaculture systems that are hard to design, they have problems with scale, or simply in simple terms, just managing proper like crop rotation, intercropping systems. So every step you do towards being one step further and being more sustainable, in my definition, you are doing a movement towards agroecology.
Hmm. And it's interesting. I don't know if you've read the science writer Carl Zimmer's essay, The Human Lake,
No, I
but
didn't.
it's a one. I'll send you a link to it, and I'll put it in the show notes. I've done so before. So what he was trying to make his readers think about is the relationship between the human body and the founding of ecology, when, oh, I cannot think of the ecologist's name, who came up with the different trophic levels in lakes and the
Hmm.
different organisms that you see at different depths because of oxygen, light, sulfide production at different levels. And they stratify into different niches, as we've been talking
Hmm.
about. And then he turns the page on you and says, but that's how the human body is too.
Mm,
And he gives
interesting.
lots of examples of this. And it's really, I like to have my students read it because it really, there are some examples that are disturbing in there, yes, but it really makes them think about that aspect, that they're not removed from the processes in nature. And I would argue it's the same thing in agriculture, but we tend to think, oh, agriculture isn't like nature, but it is indeed part of nature. It's just a different set of parameters.
Exactly. And I like to use this example that if you think about agricultural science, since we start doing agriculture, all the most like what we do today in terms of research, it is essentially research driven by monocropping system, large scale monocropping systems, right?
Yes,
So when we talk about being more sustainable or diversifying our agricultural system, some people say, well, but then I cannot harvest, then I don't have machinery for that, then I cannot manage my system. Sure, but the science was not direct to have a diversified, sustainable system, right? So the science was always driven towards a monocropping system.
right.
So we can change this perspective progressively, right? If you want to be more sustainable, because there are some resources that are finite, right? So they end eventually. So soil erosion is really removing soils faster than they are formed. So you have phosphorus, which comes from rocks. At some point, we cannot mine anymore. So there are always those things that eventually will be limited, so we need to re-envision the way we do food, or we produce basically all the food that we need. But putting another perspective, I like to have that as well. Uh, some people think about, yeah, but this large scale agriculture devastated most of the common knowledge in terms of the food system, the traditional knowledge, and there was this negative impact. But I like to think as well that was only because we have this kind of agricultural revolution that a lot of people like myself or my family could move out of the production system and work into technology and advance other areas of knowledge. There's always this balance, right? So imagine if we still need to spend half of our day around the cropping system, produce what we're going to consume. And nowadays we have kind of the beauty of just going to a supermarket, choose what you want to eat, go home and cook, and sometimes even get like cooked food, and we can invest our time, our mental kind of dedication to something else. So those debates are very interesting to put into context with students as well.
Francisco, with all the concerns that people have about global climate change in various directions, do you have any thoughts about how that might be impacting the microbiome that underlies and underpins our agriculture?
Yeah, that's a great point. The way I like to think about it is microbes, they are the ones that can produce greenhouse gases, right? But they're also the ones that can consume greenhouse gases. And going back to some points we discussed is how we manipulate the environment, either agricultural science or pollution or something, will trigger them to act in one direction or another, right? Of course, they will suffer from the consequences of that changing climate, but it's interesting to think that they are the engineers deciding the fate of climate change. And we are the people that can really rationalize and say, well, maybe that strategy or that strategy can trigger more greenhouse gas emission. Maybe that strategy can actually allocate more carbon into the soil, which they're going to stabilize and put in deep soil layers and you work as a carbon sink. So our way to manipulate the environment is really what determines that. And then microbes will respond in terms of that as well.
I really enjoy the way that you are essentially discussing the way that philosophy interacts with this. I mean, there are lots of different levels, and I'm so pleased to hear it. Um, and this is what I've seen with other people that work, and I'm going to call it agricultural biotechnology in general, is that they often do think about these things. But of course, headlines are about clicks and anger, right? So we don't hear enough about this reasonable aspect of things. Once again, tagging back on what you were saying before, Joe Handelsman's book, The Death of Soil, is very, very, very important for folks to have a look at or think about
Mhm.
because we just tend to. In fact, she would always respond poorly when people called it dirt, and
Yeah.
that's why, right? And I'm really happy to hear this. It kind of brings me back to some of the things that I was thinking about in graduate school all these years ago. Although a bunch of tubes with alfalfa seedlings isn't quite the same, it is certainly, kind of on the approach about things. And again, the interactions between different microbes and their plant partners. The work with Striga, fascinating beyond beliefs. I've been very, very happy to be able to talk to you about these things. May I ask you to talk a little bit about the path that took you to microbial sciences? You spoke a little bit about it before, but tell folks where you're from and a little bit about the path.
Yep, absolutely. So I'm originally from Brazil. My parents are not involved into any academic work. I know in America they say, well, first generation a college student. I would say myself and my siblings were first generation high school students
Mm-hmm.
because even my parents they didn't went further into their education. Uh, but my brother was the one leading the way. He's a soil microbiologist in Brazil, a professor at the University of São
Wow.
Paulo. So
Wow.
actually, as a teenager, I was just playing in a band. I was studying music and I was teaching music. And eventually, my dad came to me. I was like, "What you gonna do? You want to work or you want to study?" And I didn't realize that study is more than work, or it's both at the same time. And one day, my brother took me to the lab, and he was doing back in the day. Well, you are aware of that. We didn't have high throughput sequencing, so to kind of look into microbial diversity, we did 16S clone libraries, right? So use
Right.
a plasmid, we cloned that, and he showed me a plate with some ecolies with different kind of plasmids, and some of them worked, some didn't, and we have like these blue and white colors. And when he showed me that, I was like, "What is that?" Well, he said, "Well, I'm cloning some stuff, right?" And that was like, "Whoa, that's super cool." It's 2004, we have all this cloning story going around. I was like, and then my dad asked me again. I said, "Well, I want to do exactly what he's doing, right?" That's how I started. And then I got into agronomy, and I learned to love as I go, right? Was not something I always envisioned that, even though I was always working with, as I said, molecular microbiology and microbiology. Uh, but going back to our initial point, when I said something that you are jealous about and you're excited,
Mhm.
I always like this area of every time I see someone speaking about something, I say, I wish I could speak this way. Like I wish I could give a lecture this way. I wish I could be a good teacher. Like I think this teaching kind of aspect was something that got me early on. I was teaching music back in the day. Uh, and then I did a master's in plant microbe interactions, but at that time I was really looking for expanding my horizons, right? And then I applied for a fully funded PhD in the University of Groningen in the Netherlands. I worked with Professor Dick Van Elses and Joanna Sales, two amazing mentors. I could live 1 month in a room with Dick Van Elses in his house, and I could have dinners with him. He was talking about soil microbiology, a very humble and genius person in my perspective. He just retired. And that changed my perspective of science because even though he was a senior guy, he was excited about daily results you could have in the lab. Even showing him a gel, he did a lot of like those GGG gels to show
Ugh.
diversity. And he was super excited about it. He was like, "That's great." And I could see the excitement of science as a fun thing, right? Not as a job. Uh, and after that, I did a postdoc in there. I moved to the Netherlands Institute of Ecology, work with Jos Heijmakers, more working on plant micro interactions. Again, I was very happy to have those amazing mentors. I just met him about a month ago. And from that point, I think most of my excitement comes from really like teaching student, training students. Even though we do this grant publication craziness as a scientist, what excites me is to really see my students growing. My first postdoc got a faculty position, and to me, that was a big, big achievement. He's a personal friend. We write grants together now, and I have an amazing team here. I call them a family. And they say, "Why we are your family?" I said, "Well, because I spend 3 hours a day with my kids, and I spend 8 hours a day with you, right? So they are my family." And I try to balance things out, right? I like to enjoy my kids; they are young, and to have this kind of work-life balance as well. That's essential for me.
Oh, this sounds just exactly perfect. So many people think there's just one path, but look at all the excitement that you've talked about. By the way, I like the bit about family. We call it here hashtag LabFam. And it's good to see that group not necessarily my own involvement, but how they support one another. And again, it's wonderful to see your students succeed.
Yeah, that's the best part of our job for sure.
It is. I want to thank you very much for being on the podcast. I have so many questions. I will be writing to you with some of those questions that I have about things that I do, because you're very enthusiastic. You look at things from outside and see these interconnections, and I am really, really happy to have had you on this program.
Thank you so much. Yeah, thank you for having me and to listen to my ideas and way of kind of envision agricultural science and microbiology and their integration. It was a pleasure to be here for sure.
I really, really, really have enjoyed it. Thank you.
Thank you.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. I'd love to hear from you. Show notes from today's episode with, as usual, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Francisco Dini Andreote can be found in the Department of Plant Science at Pennsylvania State University in University Park, Pennsylvania. Many, many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.